Hello everyone and welcome to our video on positron emission tomography, which is a type of scan. First of all, I'd like to cover a bit of basic physics, particularly positron emission. First, let's look at what a normal atom is made of. A normal atom is made of a nucleus, which is surrounded by electrons. The nucleus consists of two subatomic particles called protons and neutrons. This makes up the nucleus, and the nucleus is surrounded by electrons. So I'll try to make a very basic graphical form here. So imagine the orange or the yellow is the protons, and the green is the neutrons. Green, yellow. So there's your protons and neutrons, which constitute the nucleus. And then imagine that you have little, relatively massless electrons surrounding or orbiting the nucleus. So you can have as many as you want here. However, sometimes in nature and from lab creations, we have something that are called unstable nuclei. Unstable nuclei cannot last for long. These are also known as radionuclides, because they produce radio frequencies. One example of an unstable nucleus is one that is proton rich, and this is the one that emits positrons. So how does positron emission work? If you have too many protons, the typical pathway is that some of the protons convert into neutrons. And let's go back to our first point here. Remember that protons are positively charged and neutrons have no charge. And later on it'll be important, electrons have a negative charge. So this means that in an unstable nucleus, the proton, which is positive, becomes neutron, which is neutral. Where does this positive charge go? Well, it's released as a subatomic particle, which is called a positron. A positron is literally the opposite of an electron. This process is also known in some textbooks as beta plus decay, because positrons are known as beta plus molecules, or beta plus particles. This positron inevitably collides with an ordinary electron. And because they are basically the same mass, but opposite charge, they're known as antiparticles. Equals antiparticles. And when antiparticles collide, they annihilate, which means they lose their mass and release all of their energy in the form of rays, in this case 2 gamma rays. And these gamma rays travel in opposite directions. So I'll illustrate that again here just for easier understanding. Let us say that we have an E minus, so an electron traveling in one direction. So here's your electron and it's traveling towards the bottom right corner here. However, also traveling towards it is its antiparticle, the E plus, or the positron, which was released by the unstable nucleus. And this is traveling in this direction. Let's say they actually collide. These antiparticles collide And what happens? Well, they release their energy in the form... Wait, let's draw it squiggly to show that it's not mass, but a wave. Releases their energy in the form of gamma rays. So there's one gamma ray, and there's the other gamma ray. And that is the process of annihilation. These two gamma rays are what are now important to us. So let's go back to the PET scan for a second. Before we do the actual scan, we must inject something called a tracer into the patient. A tracer is injected into patient, and whatever the tracer consists of, which I'll explain now, eventually reaches the brain. Okay, so what does the tracer consist of? It consists of two main molecules. First is what we just discussed, the positron emitting radionuclide. And second is a biologically active molecule. Biologically active molecule is just something that gets metabolized, or in even simpler words, used somewhere in the body. This is to ensure that the tracer just doesn't go in random directions. Okay. So this travels through, the positron is emitted, and then something called gamma cameras pick up the two gamma rays going in opposite directions. How do gamma cameras work? So I have the image in the bottom here to illustrate what goes on. So let's illustrate that a positron was traveling in this direction within the head, and an electron was traveling the opposite direction within the head also. They collide and they produce two gamma rays, which you can see illustrated by these squiggly lines here. There's one and there's the other. Over here is where the gamma cameras are. The gamma cameras pick up these gamma rays, and using the delays, this is an important point, using the delays in arrival on each side. So, for example, this one might be earlier, this one might be later. The difference in arrival of each gamma ray indicates where in the head it came from. So by source, I mean where in the brain it came from. So logically, if you have more rays coming from the same 3D location in the brain, it means that that brain is more active, because as we said, biologically active. That means that it is more biologically active if a source is producing more rays. Now, just to illustrate an example of a brain PET scan. A common tracer, and one which appears to be used here, is the glucose scan. The glucose, the active molecule is 18 fluoride FDG, which also is called fluorodeoxyglucose. So FDG. Again, what does it measure? It measures regional glucose metabolism, because glucose is the active molecule. Just basics of looking at a graph, like looking at this scan. Red or yellow. So red, you can see around some of the cortical areas here, something like that, and here. These red indicates high activity. Blue, on the other hand, where can I write this? Blue is low activity. So you have some areas here. Okay, so that's the color coding. But what are the normal findings? This is a normal scan, so what can we see here? Let's write this down here. Normal findings are, first of all, that the cortex, or the surface or higher part of the brain, has high uptake. Cortex equals high uptake. And then there is lower uptake. So subcortical has lower uptake, equals lower uptake. And also white matter tracts. White matter is just the areas that are myelinated or covered with a sheath. Those are also indicated with lower uptake on PET scans. So what is a PET scan used for? Well, we can use it for multiple purposes. Currently, it's not really used in screening or diagnosis. It's mainly used in research purposes, but it is useful for functional diseases such as dementia, epilepsy, cancer, and it's also being used to link to psychiatric research, psychiatric diseases.